Hello everyone, I am Oves Bagwan and my topic is pharmacodynamics. So, topics to be covered are pharmacodynamics, its action, effect sequence, transducer mechanism, regulation of receptor, dose response relationship, drug potency and efficacy, therapeutic efficacy, drug selectivity, drug synergistic and antagonism. So, let us now discuss about the first topic, that is pharmacodynamics action and its effect sequence. So, principles of drug action, stimulation. It is the selective enhancement of the action of specialized cell. Example, adrenaline stimulates heart, pilocarpine stimulates salivary gland. Second, depression. It is the selective inhibition of the action of specialized cell. Example, barbiturates depresses CNS, quinidine depresses heart, omeprazole depresses gastric acid secretion. Third, irritation. It is the non-selective and noxious effect to the less specialized cell. Irritation may result in inflammation, corrosion or necrosis to cells. Fourth, replacement. It is the use of natural metabolites, hormones, their congeners in deficiency states. Example, insulin in diabetes, levodopa in Parkinsonism. Fifth is cytotoxic action. It is the selective action on parasites or cancer cell without affecting other cells of the body. Example, penicillin, zidovudine, methotrexate. Now let us discuss about the second topic. According to the molecular structure and nature of mechanism, there are four types of drug receptor. Type 1, ligand-gated ion channel, which is ionotropic. These are the membrane protein with a similar structure to other ion channels and incorporate a ligand binding receptor site, usually in the extracellular domain. Typically, these are the receptors on which fast neurotransmitter act. Example, GABA receptor. Second type is G protein coupled receptor. These are known as a metabotropic receptor or 7 transmembrane spannic receptor. They are the membrane receptor that are coupled to intracellular effector system via a G protein. They constitute the largest family and include receptor for many hormones and slow transmitter. Example, dopamine receptor. GPCR acts by adenyl cyclase, cyclic AMP pathway, phospholipase C, ion channels. Now let us discuss about the type 3, which is kinase linked receptor. This is the large and heterogeneous group of membrane receptor responding mainly to a protein mediators. It comprises an extracellular ligand binding domain linked to an intracellular domain by a single transmembrane helix. Example, cytokine receptor. Now, other topic is dose response relationship. It is the response given by a drug once administered to the body. Dose response may vary between the different species and within same species under different conditions. Therefore, drug response studies help to minimize errors and predict the efficacy of other drugs. Each drug has a characteristic drug response curve for specified set of conditions. In general, drug response curve conforms to S-shape or sigmoid type. Other topic is regulation of receptor. So there are two different methods for regulation of receptor. First method is upregulation, which is cell increase the quantity of cellular component. Other is downregulation, cell decrease the quantity of cellular component in response to external variable. Now drug potency and efficacy. So drug potency means measure of dose required to produce a response, and drug efficacy means maximal response a drug can produce. Therapeutic efficacy, MSCs. So first is donor variation, which includes donor age, stem cell content, clinical background. Second is tissue origin, bone marrow, adipose tissue, peripheral blood. And third is cultural time, early passage, lower passage, higher passage. Fourth is supplements, autolog, allogen, xenogen and defined supplements. Fifth is cell delivery, cell dose or timing, systemic infusion and local injections. Now the last topic is drug selectivity and specificity. So drug selectivity. If an analytical procedure is able to separate and resolve the various components of a mixture and detect the analyte qualitatively, it is drug selectivity, which is restricted to a qualitative detection of the components of a sample. And drug specificity means if the method determines or measures quantitatively the component of interest in the sample matrix without separation, which is restricted to quantitative measurement of one or more analyte. And the last topic is drug synergism and antagonism. So drug synergism means it is a combined effect of a drug. When drugs are combined together, one drug may enhance the activity of other drug, or both drugs can have better effects together. And its effects are, first is additive effects. When two or more drugs are combined together, their effects are increased. Ex- So example of additive effects are aspirin and paracetamol, and second is amlodipine and atenolol. Second action is potentiation. When two or more drugs are combined and their effects are more than the additive action. Example, levodopa and carbidopa and acetylcholine and physostigmine. Drug antagonism. It is the reduced or abolished effect of drugs when combined together. One drug may inhibit the activity of another or reduce the potency of another drug. It may be physical antagonism, chemical antagonism, physiological antagonism, receptor antagonism. In receptor antagonism, there are two types: competitive antagonism and non-competitive antagonism. Competitive antagonism are reversible and non-competitive antagonism are non-reversible. Now, let us discuss about the some MCQs related to the topic. So, first MCQ is, which of the following is not type of antagonism? Options are physical antagonism, chemical antagonism, physiological antagonism, and D, biochemical antagonism. So obviously the option is D, which is not type of antagonism. Now let us discuss about the second MCQ, which is the enhanced effect of drug when combined together. Synergism, additive effect, potentiation, or antagonism. So the correct option is D, antagonism. Now let us discuss about the third MCQ. Which one of the following is correct about dose-response relationship? Option A, it's not the response given by drug once administered to the body. Option B, every drug has characteristic drug response. Option C, drug response curve is the sigmoid type. Option D, it's not a S type. So the correct option is C, drug response curve is the sigmoid type. Now let us discuss about the fourth MCQ.